Hello, everyone, and welcome to today's live broadcast, Validation of Mass Spec Analytical Platforms and Proteomic Biomarkers, FDA Perspective, presented by Dr. Doug Jeffery. We're excited to bring you this educational web seminar presented by LabRoots. For more information, please visit labroots.com. My name is Soon Pham, and I'll be your moderator for today's event. Before we begin, I would like to remind everyone that this event is interactive. We encourage you to participate by submitting as many questions as you want at any time you want during the presentation. Just click on the green Q&A button located in the lower left of the presentation window and type your question into the box that appears on the screen. We'll answer as many questions as we have time for at the end of the presentation. Also, please notice that you will be viewing the presentation in the slide window. To enlarge the window, just click on the screen icon located on the lower right. Finally, if you have trouble seeing or hearing the presentation, please click on the support button at the top right of the presentation window, or use the Q&A button to let us know that you're having a problem. Now, I'd like to introduce Dr. Doug Jeffery. Dr. Jeffery received his Bachelor of Science degree in biology from the Massachusetts Institute of Technology. He then received his PhD in biochemistry from the University of California, San Francisco. Afterwards, he joined the Novartis Institute of Biomedical Research, where he performed basic research in oncology target discovery. Dr. Jeffery is currently a scientific reviewer at the Food and Drug Administration Center for Devices and Radiological Health. Please join me in welcoming Dr. Doug Jeffery. I will now turn the presentation over to him.
Great. Thank you, Joanne, for the introduction, and thank you to LabRoots for giving me the opportunity to talk about mass spectrometry and clinical in vitro diagnostics. So here, the outline of my talk is here. I'm going to start in the first part of the talk and discuss the role that FDA plays in the regulation of in vitro diagnostics. And then in the middle part of the talk, discuss our experience directly with mass spectrometry and the use of mass spectrometry in vitro diagnostics, and then talk a little bit about the end, specifically about protein and peptide identification and quantitation using mass spectrometry. So this is the overall organization of the Food and Drug Administration. I am in the Center for Diagnostics and Radiological Health. And specifically in the Office of In Vitro Diagnostics and Radiological Health. We are one of 7 centers within the FDA. Some of you may have heard of CDER or know about CDER. CDER is the center that regulates and clears or approves most of the new drugs that are marketed in the United States. Our role in CDRH and in OIR in particular is to regulate and approve or clear in vitro diagnostics that are used to diagnose disease. Just to start out very broadly, the definition of an in vitro diagnostic according to the Food, Drug, and Cosmetic Act is an instrument, well, this is the definition of a medical device, is an instrument, apparatus, implement, machine, detritus, implant, or in vitro reagent that is intended for use in the diagnosis of disease or other conditions. In particular, our role is to regulate, in OIRs, to regulate in vitro reagents that are intended for use in the diagnosis of disease or other conditions. And what that means is that we regulate medical devices that measure, that are used to measure anything which is taken from the body. So that's in contrast to things like stents or hip implants, which are actually implanted in the body. Those are regulated by a different office within the centers for within CDRH, and we regulate only those in vitro reagents or in vitro diagnostics. The way that we review in vitro diagnostic submissions, and in fact all submissions, is outlined here. The most important, one of the most important parts of a submission for an in vitro diagnostic is to define an intended use. So this essentially describes how the IVD is meant to be used, for screening for disease in the population, for prognosis, or other things which we'll go into later. The other important thing is to understand what patient population is meant to be tested with this device. For example, it's patients that have or are suspected to have rheumatoid arthritis, or patients that are may undergo treatment for a particular disease, and an IVD might be used to decide which treatment they get. The intended use defines what we call the risk of the device, and the risk is, there are 3 classes of risk, from low to high risk. And the risk of the device, in turn, defines which type of submission that is required at the FDA to gain clearance or approval for marketing of that device in the United States. There are the 2 major types of device are PMAs, or pre-market approvals, for the highest risk Class 3 devices. And then moderate risk or Class 2 devices are cleared through the 510K process. We'll talk more about this in a minute. Just to emphasize and we'll talk about this in more detail, a classification, either Class 1, 2, or 3, determines the scope of the studies that are necessary to demonstrate the performance of the device, both the analytical and the clinical performance. What's not shown up above, but is important, and this will be the only time I mention it, is using a device or measuring a particular biomarker in a clinical trial may require what's called an IDE, or an investigational device exemption. And so if anyone is interested or needs to use an IVD or a particular assay in a clinical trial, you would need to contact the FDA and go through the IDE process. Lastly, what's underlying here, and we'll talk about this at the end, is the pre-submission process. So this is something that is accessible to anyone, and it's basically an opportunity for device developers, device manufacturers to submit questions, submit a description of your device and questions to the FDA to help guide the development of the device. And we'll talk in more detail about that at the end of the talk. So one of the key things that we we regulate or we're interested in understanding about devices is the safety and effectiveness. And what do we mean by safety and effectiveness? This is sort of a broad brush look at it, but there are sort of 2 main components of safety of a device. One is how accurate are the results of the test. So if you're measuring a particular analyte in, say, serum or plasma, the analytical performance needs to be tested, and studies need to be performed to look at, for instance, the accuracy of the device, the linearity, the precision, and the reproducibility of the device, and we evaluate the analytical performance to determine whether the device actually is able to measure reproducibly and well the analyte that it's intended to measure. The other important piece of safety is what is the risk to the patient of a false positive or false negative result. Basically, what happens if the test is wrong, and what's the potential outcome in terms of the patient's health or safety. 2 simple examples of this are an immunoassay that measures rheumatoid factor could be used, is often used as an aid in the diagnosis of rheumatoid arthritis in conjunction with other symptoms or patient history. So in that case, that's a moderate risk device, in the sense that if the result were wrong, there is other information that the physician and the patient are using to determine whether the patient has rheumatoid arthritis and what a potential course of treatment might be. In contrast, a high-risk device, such as one which was intended to determine what treatment a patient with, for instance, cancer might get, if that device were to be wrong, that patient might not get potentially life-saving or life-prolonging medication, or alternatively, if a patient might get medication that they don't need and might have significant side effects and consequences for their health. So the risk to the patient of a false result is something that we take, that we consider very seriously. The effectiveness part is basically asks the question, is the test relevant to the clinical condition? This is done by performing clinical studies and looking at the clinical performance of the device. So as I said before, the risk classification, either Class 1, 2, or 3, determines the type of submission that's required. So on the left-hand side, you can see Class 2 devices require, and some Class 1 devices require what's called a 510K pre-market notification. And then Class 3 devices require a submission which is called a pre-market approval. And both of these submissions are required before a device manufacturer or developer can market the drug in the device in the United States. There are some pretty significant differences between these 2. As you can imagine, a Class 3 high-risk device requires more clinical, more evidence, more valid scientific evidence. It always requires clinical data It requires a lengthy review process, including manufacturing inspections, whereas a 510K device is cleared by comparison to an existing predicate device. So there are many, let's take an example, immunoassays. There are many immunoassays that have been cleared already and are marketed to measure something like rheumatoid factor for rheumatoid arthritis. And so a new device that measures rheumatoid factor would then be compared to an immunoassay or a device that already measures that analyte, and the performance of that device has to be equivalent to the predicate device. The 510K submission is a little bit shorter. There is a manufacturing review, and the requirements to get marketing approval are somewhat less than for a PMA. So I wanted to talk a little bit about intended use. This is a very important thing that which defines the risk of the device, as I mentioned before. There are a lot of different potential broad intended uses that are described here. I'm not going to go through all of them. So sort of talk generally about screening and diagnosis. So a screening intended use would be one that would aid in infectious disease with patients that have no signs or symptoms, so they seem otherwise normal. The diagnostic intended use would be aiding diagnosis of patients who already have signs and symptoms. For instance, the different uses of the different intended uses, types of intended uses have potentially different risk determination classifications. So, for instance, a screening device would potentially be class 3 because you're talking about normal subjects, people who are otherwise healthy, and if the test is wrong, you might miss those people who actually have disease or patients who don't have disease, and they might get some unnecessary treatments. A diagnostic claim is class 2 or class 3 potentially. I'll keep using the rheumatoid arthritis example, where a rheumatoid factor test would be used in patients who already have signs and symptoms of rheumatoid arthritis, and the risk there is, of course, that if the test is wrong, you might not treat them appropriately, and if the test is, or you might give them treatments that they don't necessarily need. An important thing for consideration of intended uses is defining the population of patients, like I talked about at the very beginning. And so in this example, sort of thinking about this very generally, a screening device, which is meant to be tested in normal, healthy population, would require that type of patient in their clinical studies that are being done. It could be done in a normal, healthy population that it's at risk for disease. For example, a lung cancer screening test might be used in patients of a certain age who have a history of smoking but are otherwise healthy and have no symptoms. A diagnostic intended use would use patients who already have the signs and symptoms. So it's very important that these studies are not done in a normal population, but rather patients, in the case of rheumatoid arthritis, who have some joint pain or difficulty, basically joint pain. So in terms of the analytical studies that are required for 510Ks, there are a number of different sources of information for designing studies. Talk really in terms of the way these sources of information are used actually in the development of tests and the kinds of tests that we require in the submissions. So I'm not going to talk too much about the column on the left, which is the ICH, which is mostly for drugs and biologics. But the two that are sort of important for in vitro diagnostics are CLIA and CLSI. So the CLIA regulations are part of the Center for Medicare and Medicaid Services. They're intended mostly for clinical labs and ensure the proper training of the lab personnel and the performance of the assays. The CLSI documents are something which FDA and we at CDRH recognize as important guidelines for the development of IVDs. They're intended both for clinical labs and device manufacturers, and they provide an excellent some of them, the ones that we recognize, provide an excellent source of information for study design and data analysis that can be used for the analytical validation of IVDs. And the CLSI documents that we recognize at CDRH are all available publicly on the CDRH website. Right, I think I talked about that. So one of the things, questions that we often get, particularly about analytical validation of in vitro diagnostic devices and determining analytical performance, is questions about acceptance criteria and how does the device developer know what acceptance criteria are okay for their particular device. So a simple example of acceptance criteria would be when looking at the precision and reproducibility of the device, what's an acceptable percent coefficient of variation or percent CV when measuring the same sample over and over again. And there are acceptance criteria for all of the analytical performance testing that is done, but that's just one example. So the challenge here is that the acceptance criteria and study design depend on the type of the device, the technology behind the device, and also the intended use. So you could imagine, I think, pretty easily that the acceptance criteria for a genotyping test versus which uses next generation sequencing or PCR would be very different for an immunoassay that's used to measure autoimmune disease. And some of the other examples of different types of devices by the technology and intended use are listed below. So it's very difficult for us to define acceptance criteria. It depends really on the use of the device, the population of patients that are being tested, and the type of technology that's being used. This is just sort of a simple example of, sorry, get rid of that. Ignore that red arrow, sorry. This is a simple example of two different types of devices that I mentioned in the previous slide. The first one would be genotyping for factor VIII, mutations in factor VIII gene. The other is an immunoassay that's used to measure autoantibodies for detection of lupus in this example. And so you can see the acceptance criteria, if you just sort of go down the chart, are quite different, so particularly in the sensitivity. The sensitivity of genotyping test, the acceptance criteria for sensitivity is 100%, whereas for these autoantibodies can be as low as 24%. The study design is also very different with the number of patients that are required to perform these studies, as well as the precision. So you can see that depending on the type of test and type of technology that's being used, the acceptance criteria and the study design can be very, very different. And it's difficult a priori for us to define what those acceptance criteria should be before we understand the type of test, who it's the patient population that it's going to be used in, and also the technology that underlies the test. Some of the surprisingly common mistakes that we see when people make submissions for in vitro diagnostics, we talked about acceptance criteria. Sometimes manufacturers will set acceptance criteria, and then when the data are analyzed, they don't meet them. This is obviously a problem because data that don't meet the acceptance criteria cannot be used to verify or validate performance of a device. Another important one is not performing the analytical and clinical validation on the final device. So sometimes manufacturers will change the device after they've done their data collection and data analysis. It's obviously the performance of the device, the performance data that we receive has to be done on the final device that would ultimately be cleared for approval. And I'm not going to go through a lot of these. You can read some of the other surprisingly common mistakes that we see when people submit data for in vitro diagnostic devices. Talked about the different types of submissions, mostly 510Ks and PMAs, and Also, some very basic things about intended uses and analytical and clinical performance data. A really great database for people who are interested in developing devices or manufacturing devices is the 510K databases and the PMA databases that are publicly available. This is just a screenshot of an entry for the newborn screening test using mass spectrometry, and what this arrow is pointing to here is basically a link to what's called the decision summary, which provides a general overview of the data that was submitted to clear this device, both analytical and clinical performance data, and this database exists for, an entry like this exists for every cleared or approved device that the FDA has cleared or approved over the last, since 1976. And it's an excellent source of information for people who want to, manufacturers or device developers who want to develop an IVD that is similar to an existing device. You can look at the data that was required to, in this case, clear this newborn screening test and the acceptance criteria, and et cetera. So it's a good publicly available source of information about how FDA reviews these certain types of devices and the information and the data that was used to clear it for marketing in the United States. So now I'm going to talk, so that was sort of introduction to how we here in CDRH think about in vitro diagnostics, some of the broad descriptions of intended uses, and some of the common problems problems that we see that people have when they submit their devices. I want to talk, for the rest, about specifically about mass spec and our experience with mass spectrometry in vitro diagnostic devices. We do have some experience clearing these devices, and in the last part of the talk, like I mentioned in the introduction, talk specifically about protein and peptide mass spec in vitro diagnostic devices. To start with, the mass spectrometer, the instrumentation itself, and this is also true of high-performance liquid chromatography, or HPLCs, the instruments themselves are what's called Class 1 exempt, which means they're low-risk devices that do not require a formal submission to the FDA, but they do have to be produced under good manufacturing practice, and also the manufacturers of the HPLC or the mass spec must be registered and listed, and there is also a database of companies that are registered and listed to market HPLC and mass spectrometers for clinical use, and that database is publicly available on the FDA website. The important thing to keep in mind, though, is that even though the mass spectrometer itself is Class 1 exempt, when the assay is added onto the instrument, then that whole system, the mass spec plus the assay, becomes classified as the risk of the assay itself. The risk is determined by the assay. So, for instance, a test which is used to, like rheumatoid factor, but not for rheumatoid factor, a test that is used to determine, as an aid in diagnosis of rheumatoid arthritis would be a Class 2, if it used a mass spec, and then the mass spec the mass spec itself would also be reviewed as a Class 2 device, and there would have to be a submission specifically about the instrumentation. I'm not going to read this. This is the definition of a mass spectrometer according to the statute that's in the Code of Federal Regulations. There's also one for high-performance liquid chromatography that you can search for on the database, the FDA website database. In the last little more than 10 years, the FDA actually has cleared or approved 3 different types of mass spec-based devices. The first one that I will discuss is the device to measure inborn errors of metabolism, or a newborn screening test. This is a test which is commonly done across the United States. There were several devices, three different 510Ks, which are listed here, which were basically describing targeted mass spec assays for specific metabolites, amino acids, and low molecular weight substance to identify inborn errors of metabolism. The other class, the second class, is therapeutic drug monitoring. There's been one 510K, which has been cleared to monitor the immunosuppressants, are tacrolimus, and again, this is an LC-MS-based device. And the third one, which was cleared more recently in 2012 and 2013, are devices which use MALDI-TOF protein profiling to identify, for identification of microorganisms that are part of an active infection. So just a little bit to give you some idea of the scope of the different technologies and types of studies that were required for clearance of these devices. This chart sort of outlines broadly, how the assay was defined, the procedure that's used to generate the result, as well as some of the analytical factors that were required for clearance of the devices. And the point of this slide is really twofold. One is to give you, an overview of what these devices do, and the second is to indicate that we have experience here at the FDA with regulating different mass spec technologies, whether it's LC-MS or MALDI, and also use of mass spec for the monitoring of different types of analytes in either chemical in nature in terms of therapeutic monitoring, or metabolites, or even proteins in the case of the MALDI-TOF device. I'm not going to go through this slide in a lot of detail. I think you can read some of the highlights there. What I want to focus on is some of the analytical factors, which you can see are quite different. So we can start, I think, with the MALDI-TOF on the right. This is a device which uses a colony from the microorganism, which is spotted on a plate. And some of the important things that were required to clear this device were understanding the effect of the age of the colony, the stability of the colony on the plate, what kind of culture media was used to develop the sample for the spotting. So sort of very device-specific and analyte-specific questions that needed to be addressed by the manufacturer before clearance of this device. The other two devices were more similar in the sense that they're LC-MS devices that are used to quantify chemical compounds or metabolites. For the newborn screening assay, an important component of it was actually the sample prep. So this is uses a dried blood spot from a heel stick from infants, and so there was some validation of essentially the sample collection reagents. And both of them also, of course, had analytical, had analysis of the extraction efficiency, the carryover, reproducibility, et cetera. So again, just an overview of the three different types of tests that have been cleared by the FDA so far that use mass spectrometry as the technology platform for in vitro diagnostic. So now I want to finish the talk, finish the section about mass spectrometry, in particular talking about protein and peptide mass spec, which is of particular interest to me. I think that probably many people on the who are attending this online know that there have been, over the last 15 to 20 years, a large number of proteomic discovery experiments to try to identify biomarkers that could be used as in vitro diagnostics, and this could be either using mass spectrometry as the technology platform, or antibody arrays, or other proteomic technologies which are designed to quantify or identify proteins on a large scale, on omics scale. Unfortunately, none of these discovery experiments that have been done over the last 15 or 20 years have translated into an FDA cleared or approved in vitro diagnostic that uses mass spectrometry as the technology platform. And there could be a number of reasons for this. The instrumentation is quite complex and potentially difficult to transfer into a clinical lab. The sample preparation is quite complex, which we'll talk about in more detail. But lately, there have been efforts to improve the reproducibility in the workflow of papers that have been published in the literature, specifically looking at peptide and protein, specifically looking at peptide mass spectrometry as an effort to try to move peptide and protein mass spec closer to the clinical lab. And there are currently manufacturers and clinical labs that are starting to offer mass spec-based assays as laboratory developed tests for use in diagnosis, clinical diagnoses. So one could ask, why is it that mass spectrometry has not been very well adopted for peptide and protein mass spectrometry for use in the clinic? It is a widely used platform for metabolites and chemical compounds. We talked about newborn screening and therapeutic drug monitoring. It's also used quite often for looking at drugs of abuse. And that's because it has, as many of you probably online know, that it has can have a very high sensitivity, good precision. I think one of the keys here for mass spectrometry, which is different from other technologies, is the ability to multiplex. The newborn screen is a good example of that, where more than 30 different compounds can be analyzed from a single sample. The other nice thing about mass spectrometry is that the assays can be constantly updated and optimized, and new assays can be developed rather quickly once if a clinical lab has expertise in instrumentation in mass spectrometry. So the question is, why is this not translated more into peptide and protein mass spectrometry? And we believe that the experience, the decades of experience that manufacturers and the FDA has with clearance of immunoassays as in vitro diagnostics can be applied to peptide and protein mass spec as sort of highlighted or discussed very briefly in this next slide. So in many ways, we believe that mass spectrometry-based assays are just like every other technology. So in this case, we think about an immunoassay as a similar type of in vitro diagnostic to measure a peptide or a protein. The analytical validation that's required is the same, whether you use, say, an immunoassay or mass spectrometry, linearity, precision, reproducibility, et cetera, the things that we talked about earlier. Clinical validation should not be any different. The test, whether it's an immunoassay or a mass spectrometry-based device, has to demonstrate effectiveness, clinical effectiveness. And basically, the goals and the data that are required to demonstrate analytical and clinical performance are no different for mass spec than they are for any other type of device. However, as I'm sure many of you know, there are differences. There are other challenges that are associated with mass spectrometry-based devices, particularly those that are used to measure peptides and proteins. The sample preparation can be quite complex. So, for instance, a serum or a plasma sample might be depleted of the most abundant proteins to try to increase sensitivity. For peptides, of course, there's a trypsin digestion step, which can cause variability. Sorry about this type, which should say immunopurification. Some tests that are being developed, certainly in literature, are either immunoprecipitating the protein before analysis or immunoprecipitating the peptide that's being measured by mass spectrometry. And that creates a complex workflow that is different from the immunoassays that are typically used to measure proteins. The other thing, the sort of great advantage of mass spectrometry is the ability to multiplex, but this creates additional complexities, both analytically as well as in the evaluation of the results. If, for instance, if the multiplex result is meant to be combined, if the individual multiplex results are meant to be combined into a single score through an algorithm, that can create complexity that is not common in immunoassays, but certainly not something which we're not, it certainly is something we're used to dealing with and evaluating here at the FDA. And as I said before, one of the advantages of mass spectrometry is the ability to develop very quickly methods to measure novel analytes. So this is not as easy to do with immunoassays because of the lack of available antibodies. But one of the challenges with developing many different methods for many different novel analytes is that there are not gold standards or reference methods for those analytes, and that can create analytical performance challenges for device developers, device manufacturers. Just to dig down a little bit deeper into sample preparation and sample and reagent stability, one of the things which is likely is with a complex workflow that involves a generation of, say, peptides for a peptide mass spec-based assay. The sample collection, the sample digestion, the sample cleanup, et cetera, can create a lot of variability, and there would most likely be a need to evaluate the effect of sample preparation at multiple sites with multiple users, multiple instruments, if it's an automated sample preparation assay. Additionally, because these workflows are so complex, instead of taking 20 minutes to an hour to complete to get to the result of the test, as is the case with many automated immunoassays, the process could take many hours or perhaps even several days to go from collection to sample preparation, and there may be stopping points along the way where samples need to be stored, and the sample stability would need to be evaluated at each of those stages to understand how storage and preparation over time is affecting the stability of the analyte and the result of the test. Sorry, ignore that arrow. Okay, we'll go on to the next slide. The other really important thing in our view in terms of peptide and protein mass spec, and this is also applies to many of the metabolite and chemical compound analysis using mass spectrometry, is the choice and use of internal standards. One of the great advantages of mass spectrometry is the ability to use a isotopically heavy labeled internal standard, which allows for a very high level of specificity and certainty that the analyte being measured is the correct one, as well as the ability to normalize within a sample and a process sample to improve reproducibility. The challenge is for device developers and manufacturers, as well as for the FDA, is what sort of internal standard would need to be used. A peptide or a whole protein, is it possible to use a housekeeping protein as a way to normalize, or perhaps spiking in a standard protein? Another question of interest is when to spike. So ideally one would spike at the very beginning before any sample processing has happened. That may not be possible. And then there also may be questions about monitoring trypsinization and the reproducibility and the robustness of trypsinization, so there might be several standards that could be added at different parts of the process. Don't have any answers to these questions right now. It really will depend on the type of test and the precision and the reproducibility that's required, and the effect of using these internal standards at different points in the workflow to make sure the test is robust and has good analytical performance. So another thing I talked about before is the fact that mass spec assays are often multiplexed or can be multiplexed, and many device manufacturers and developers are interested in taking advantage of that. We do have experience at the FDA with that. We call this an IVD multivariate index assay, or IVD-MIA, which is basically measuring several analytes in the same assay. The question will come up, how much evaluation of each individual feature is needed. In this case, it would say you were measuring 3 different peptides from 3 different proteins and combining them into an algorithm, for example. Obviously, with any individual peak, and certainly with multiple peaks, the variability of the peak is a concern. I think probably the most important part of this slide is really the last bullet point here. When it comes to understanding which peaks are most important and how much evaluation of each individual feature is needed, a developer or manufacturer should ask which features are key to making the clinical decision. So those are the features that need to be understood in the most detail from an analytical performance. And then of those features that are key to making a clinical decision, which are most sensitive to the assay conditions, and which assay conditions are most affecting the variability in the detection and quantitation of those peaks. The point of the slide is to sort of point out not only these questions, but also to reemphasize that this is something that we're very familiar with in terms of devices at the FDA, and so a mass spec-based device that came in with that measured multiple analytes would be something that we're very comfortable with evaluating. If an important point to make, and I won't go through all the bullets in this slide, but an important point to make is that any IVD MIA that takes the result of each individual feature and combines into an algorithm is considered, the algorithm itself is considered a medical device, and there is a lot of guidance available for how to, the kind of information that needs to be submitted in terms of software that's contained in medical devices. And so don't have time to go into a lot of this detail here, but recommend that the people review those guidance documents to understand how algorithms are reviewed and the kind of data that needs to be submitted. So the last two parts of the talk are about how you people can interact with the FDA and how we interact with the community. So over the last 5 years, we've done quite a bit of outreach, in particular to the proteomics community, to help try to build a clear and useful regulatory pathway that device developers and manufacturers can follow to submit a mass spec-based IVD. We've been working with this community for some time, like I said, for more than 5 years. And there's some resources there at the end of the talk, and the last few slides in the talk that are available to help guide assay development. But the first thing I want to talk about is in 2010, there was a workshop with the NIH and FDA and many other people who look at the analytical validation issues that are specific for multiplexed proteomic platforms, and both mass spec-based, and there was also an SNE-based platform. The results of this workshop were published, and there's the reference. You can see it right there. One of the things, the really important things that came out of that workshop was something called a Mach 510K. It was a further collaboration between the FDA and the NIH and several scientists to create what we call the Mach 510K. The one that's sort of most relevant to this discussion is a Mach 510K describing a multiplex multiple reaction monitoring mass spec platform. And basically, and this of course was also published, and the references are here, but the bottom line was that people worked with the NIH and FDA to submit and describe a device that measured multiple peptides using MRM and mass spec. These were submitted to the FDA, not officially sort of filed in quotes, and the FDA made comments on it, on the submission, as a way to try to help device developers, manufacturers understand the kinds of questions that the FDA would ask about such a device, and encourage you all to read those papers. Some of the typical comments that came out of the submission, this Mach 510K submission, are very, I'm not going to go through all these, but the concerns were frequently found in other types of submissions. So again, emphasizing the fact that mass spec devices are not, are in many ways very, very similar to other devices in terms of the kinds of questions and concerns that the FDA has in terms of analytical and clinical performance. And finally, about 1.5 years ago now, in June of 2014, my colleague Julia Lathrop organized a public workshop on proteomics in the clinic. There were many speakers. It was a day-long conference discussing the scientific and regulatory challenges surrounding validation of proteomic methodologies, including mass spectrometry. And if you're interested in reading a transcript as well as seeing video of all the presentations, you can go to that link to see what happened at the public workshop in June of 2014. So finally, I want to end in the last couple of minutes talking about the pre-submission process. This is another way for device developers and manufacturers to interact with the FDA. This is a formalized process at CDRH. The idea is that device developers and manufacturers can contact the FDA early and get feedback on the types of devices, the sort of technology behind the device, the analytical and clinical testing that is being proposed. I think the key thing is it's free, so anyone can submit a pre-submission to the FDA to discuss their in vitro diagnostic device. Another key point is that it's non-binding, which means that the FDA is happy to provide advice and recommendations about how a particular IVD might be, the studies that might be used to validate the clinical and analytical performance. But of course, it's not meant to be an approval process. So the approval process has to be done through a formal 510K or PMA submission. Basically, the FDA review team will look at the device and spend quite a bit of time, look at the documentation about the device, spend quite a bit of time providing feedback to those specific questions, perhaps more general comments about the device and how a device developer might think about doing the studies and generating the data for a submission. And if you want more information about the pre-submission process, there's a link here to a guidance document which gives quite a bit of information about the process and how it can be used by device developers. So in conclusion, as I said at the beginning, the intended use of the device is the most critical parameter. So is it meant to be a screening or a diagnostic or meant to guide treatment, et cetera, and in which patients. So the intended use includes both how it's meant to be used and also in the patient population. That guides not only the risk of the device, but also the studies that are required to show the clinical and analytical performance of the device. One of the critical things that we look at in the FDA is what happens, what's the risk of device failing, and so understanding the failure modes of the different test systems will also direct the focus of clinical and analytical validation for testing that needs to be done. We are very interested in mass spec-based devices, particularly for peptides and proteins. Of course, for chemical molecules, chemical compounds and metabolites as well. But I focused on the end about peptide and protein mass spec. We're very interested in helping device developers and manufacturers understand how to bring those devices, such a device, to the FDA for clearance or approval. And finally, you can talk to the FDA either through the pre-submission process or through our outreach efforts. We go to conferences around the United States to meet manufacturers and device developers and talk to them about the particular problem. So if you have any questions or comments about this talk, I'm happy to if you would email or call me. And that's the end. I want to thank, in particular for this talk, my colleagues Julia Lathrop and Liz Stafford, who are in the Division of Immunology and Hematology Devices. And I will end there. I'm sorry, one more thing there. Here at the end, the two slides, some of the references that I talked about for FDA guidance documents, the Mach 510K presentations, as well as some FDA resources here that are specifically related to mass spectrometry-based devices. So I'll leave it there, and happy to answer any questions.
Well, thank you, Dr. Jeffery, for the informative presentation. I think we all learned a little bit more about the FDA process for devices. It's time now for Q&A. So if you have a question that you'd like to ask Dr. Jeffery, please do so now. Just click on the green Q&A button at the lower left of the presentation window, type in your question in the box that appears on your screen, and click the send button. We'll be happy to answer as many questions as we have time for. So we'll give you guys a little bit of time to put in your questions. But in the meantime, I'd have a question of my own to ask you, Dr. Jeffery, if you don't mind. So I'd like to know if you can comment on the process by which the FDA grants the expedited access pathway status for a device and what that means for the review process of that device.
That's an excellent question. I would have to point you to the guidance document. I think that it's very clear from the guidance document on the EIA expedited access, EAP. The idea is that similar to the program in CEDAR, devices that are for diseases that are of sort of a high unmet need, or where other devices don't exist already, are candidates for the EAP program, and the EAP program would give sort of a higher level of access to the device manufacturer and more resources on the FDA side to try to expedite the clearance or approval of such a device for marketing. There are specific criteria that have to be met by the device manufacturer in terms of the type of device, the intended use population, et cetera. I don't have the obviously I don't have a reference on the slides to the guidance document, but if you go to the FDA website, there is a guidance document for the how to get access, how to apply for the EAP program, and the types of devices and the type of information that's required to apply.
Well, thank you so much for that. We have another question that says, if in the same mass spec I do more than one assay, it should be validated as a different system with every new assay?
Sorry, can you repeat the question?
Yeah, so the question is, if in the same mass spec the person does more than one assay, should it be validated as a different system with every new assay?
I think the question is, if an instrument is cleared for one assay, does it then have to be cleared again for the second assay? And I think it's going to really depend on the two different types of assays that are being cleared. So, like we said, each assay is evaluated separately in terms of its risk, whether it's a class 1, a class 2, or a class 3. So, for example, if a class 2 assay were cleared on a particular instrument, and then a class 3 device would want it to be, a manufacturer or developer wanted to clear a class 3 device on that same instrument, that would be a completely new submission, and the instrument would have to be evaluated in the context of the class 3 assay. So it really depends on the risk of the risk classification of the assays that are being used on a particular instrument system.
Okay. Another person asked if you could comment on the patentability of mass spec-based tests.
Unfortunately, I cannot. Here at the FDA, we don't look at or evaluate patents in any way. That's a more of a business or scientific aspect of device development. It's not something that we consider in any way.
Okay. Mark or Maheen wants to know if there's a difference between high resolution MS and MS/MS. I'm not sure what the MS/MS is.
Yeah, no, I do. So the question is about the different types of instrumentation that's available for mass spec researchers and clinicians using high resolution instrumentation, such as perhaps maybe a question. I'll just speculate that it might be about, say, a Q exactive instrument versus a standard triple quad that does tandem mass spec. Right now, it's hard to say, particularly about the high-resolution mass spec. I think that both the community and the FDA have a lot of experience with a more standard triple quad MSMS system for use in clinical IVDs. The high-resolution instrumentation can be used, obviously, in that mode, in MSMS mode. But it's not commonly used. So at this point, it's hard to say. I don't think there's enough information out there, certainly not in terms of IVD use, to highlight any obvious differences between the two. It really is going to depend on, again, the type of assay, the intended use population, and whether the high-resolution data is providing any information that sort of a typical MSMS, a tandem MS using a triple quad would not provide, and what the impact is that on the clinical, on the outcome of the test, and ultimately the clinical decision that's being made. So I think at this point, in my opinion, it's a little too early to be able to say whether there are any significant differences between the two. I think it requires more studies and more data from the use of the high resolution mass spectrometers in the clinical labs.
Okay. Erin would like to know. She says, "I thought that LDTs are currently exempt. Are these procedures based then on the proposed guidelines?"
So I would recommend, she's referring to the proposed guidance that was developed by the FDA for regulation of laboratory developed tests. This talk has not meant in any way to provide comment or information about the regulation of LDTs. But in fact, yesterday there was a discussion, there was a talk given by the FDA by Dr. Catherine Serrano about the current status of regulation of laboratory developed tests, or LDTs. I can't comment at all about that regulation, and this talk is not meant in any way to provide more information about the guidance of regulation of LDTs. But I highly recommend that you read the proposed guidance, as well as perhaps go and listen to Dr. Serrano's talk, which she gave as part of this LabRoots webinar yesterday.
Okay, great. And so we have time for one more question. Ethan asks, what is the FDA position on multivariate diagnostic devices? For example, MS on tryptic digest of protein mixtures.
What is our position? Our position, it's a great question, and certainly we appreciate the power of multiplexing and the fact that it's most easily done using mass spectrometry. Our position is that we think it's a powerful application of the technology. We certainly have experience with multiplex assays using other technologies, and we have methods, we have processes in place to evaluate such a device, and so we highly encourage people who are developing multiplex assays to come in and either in the pre-submission form or in a formal submission and submit such devices. We're happy to provide guidance in the form of pre-submission about how such a device would be evaluated. The potential is tremendous. So all I can say is that we encourage people to submit such a device, and we're happy to discuss about a particular device, how a particular device would be evaluated here at the FDA.
Well, that's good to hear. So I would like once again to thank you for your presentation. Do you have any final comments?
I also thank thank you for moderating, and I thank LabRoots and for the folks that have been participating online. I hope that you've learned something in general about the FDA process for regulation of in vitro diagnostics, and encourage all of you who are interested, particularly in mass spectrometry-based devices, to contact me or make a pre-submission about your device. We're always happy to talk with manufacturers and device developers about these types of devices, and hope that we can help work together to forge a pathway for the regulation of these devices in the future.
Well, thank you once again, Dr. Jeffery. Before we go, I want to let everyone know that today's webcast will be available for on-demand viewing for 6 months from today's live event, which is Thursday, November 12th, 2015. You will receive an email from LabRoots letting you know when this webcast will be available for replay. Please share that announcement with your colleagues who may have missed today's live event. That's all for now. Thank you for joining us, and we hope to see you again soon. Goodbye.